I'm going to be talking today about some geometry on spaces of phylogenetic trees. This is very much joint work with Stephan Huckemann from the University of Göttingen, his PhD student Jonas, and my PhD student Maryam. So I'm very sorry not to be present in person with you in Paris. Unfortunately, I'm locked up at home, self-isolating because of COVID-19. So let's start off then by looking at a picture of some phylogenetic trees. Phylogenetic trees show evolutionary relationships between sets of present-day species. So the present-day species form the leaves of the trees. The internal vertices correspond to speciation events. And you can see I've drawn three trees with the same set of leaves, the same set of species. So there's three different alternative evolutionary histories here. You can see that the trees are edge-weighted. They have an edge length, and the length of a given edge represents a degree of evolutionary divergence. And hidden away, there's this little length scale that enables you to measure off lengths of edges, and I'll say more about that later. So you can see that these three trees differ in terms of their edge lengths, but also in terms of their branching structure. So if you look at, say, yeast down here, it's grouped with this thing C-squirt, whereas here on this tree, yeast is an outgroup. So evolutionary trees like this are very important in molecular biology. They're usually inferred from data sets of genetic sequence data. And when you do that, very typically what you end up with is not just one tree, but a whole sample of possible trees all on the same set of leaves. So for example, you might obtain hundreds or thousands of samples from a Bayesian posterior or a bootstrap procedure. What I would like to do is characterize such samples and do statistics on these samples of trees. So answer questions, simple questions like, well, what's a mean tree? How do the data vary around the mean, for example? Now, here's a little summary of what I'm going to be speaking about today. The first thing then is the Bellera-Holmes-Votman tree space, or BHV tree space. Now, I think that this is the state of the art, really, for analyzing data sets of phylogenetic trees. However, we're going to take a slightly different viewpoint that's going to take its basis in gene sequence evolution on trees. So we're going to introduce probabilistic models of gene sequence evolution, and the idea is that we can use these to map trees to distributions. Then, given a metric on a set of distributions, you can pull this back to give you a metric between trees. That's the real fundamental idea of how we obtain our new geometries. Now, I'd really like to stress the fact that the geometry we obtain is not merely a new metric on BHV tree space. It amounts to a different point set of trees. So instead of just having trees, we actually consider a set of forests. Moreover, the tree space we obtain, or the space of forests, has a different topology to the BHV tree space. Along the way, I'm going to show you that the geometry that we obtain, very roughly, is robust to changes in the assumptions about these underlying evolutionary models. There are more details about that in the paper that's associated with this talk. Okay, so let's fix some notation then. A tree is a connected acyclic graph. I always say that in my talks because otherwise people actually think I'm talking about plants, but I'm not. I'm talking about mathematical trees. And all of the trees we're going to consider have the same set of leaves, or they have labelled leaves from 1 up to n, and a root which is labelled 0. So they're going to be rooted trees. They're going to be edge weighted, so each edge has a weight which is strictly positive. So there are two pictures of two example trees underneath here. N equals 4, and we've got two different trees there. You can see the roots labelled 0. You can see that that each tree contains at most 2n-1 edges. That's if we ignore that little stubby edge leading to the root. We've got 2n-1 edges in this tree. That's because it's a binary or bifurcating tree. But we also include in our analysis trees which might have trifurcations or higher degree vertices within them, like this tree on the right. So the BHV tree space on n leaves is the set of all such rooted, edge-weighted trees. Now that's the space as a set. What Bellera, Holmes and Votman did was they showed how to embed this space in a high-dimensional Euclidean space, and they obtained a metric on the space of trees which is induced by the Euclidean metric on Rn. Now what this boils down to is that if you have two trees with the same branching pattern, the way you compute the metric is just to write down a vector of edge lengths for these two trees, and you just compute the usual L2 or Euclidean metric between these two vectors. Now, the BHV tree space is a truly remarkable and beautiful thing, and here's why. It's a cat-naught space, which means it's globally non-positively curved. And as a result of this, there's a unique geodesic, or shortest length path, between any 2 points in the space. In addition, it was later shown that there's a polynomial time algorithm for computing these geodesics, another really beautiful paper. So these two ingredients, uniqueness and existence of geodesics, plus the fact you can compute them relatively quickly, mean that you can do an awful lot of statistics on BHV tree space, such as computing means and doing analogues of PCA. But where does this assumption of the locally Euclidean metric come from, and what implications does that have? Now, questioning that assumption is what's led to these novel geometries which I'll be describing today. So our starting point is to think about evolution of genetic sequences over trees. So here's just a very quick cartoon picture of how you might imagine this occurs. Let's take three species, human, chimp and mouse. Imagine we know their evolutionary tree and think about the evolution of some DNA sequence from their ancestor to the present day. Now, what happens over time is that DNA picks up mutations, and so we end up with a certain set of mutations in human, a slightly different set of mutations in chimp, and an even more different set of mutations in mouse. And then we observe our present day genetic sequences in the three species. Now, the way we model this mathematically is using a continuous time Markov process. We're going to model not an entire sequence, but just a single site in the DNA, so just a single locus. Given a tree T, capital T, then we let xt be this Markov process, so this is the letter at a point little t in our tree. So it's going to take values in the DNA letters, or potentially we might consider simpler Markov processes just with a two-state parameter space, two states, state space. So the rate on phylogenetic, on these Markov models is generally fixed, so the edge lengths on trees represent the expected number of letter changes. This is a really crucial fact. Think back to that initial picture with that little bar indicating a scale for our trees. If we have edge length 1, then that means we expect on average each letter to have changed once over the duration of that edge. Okay, so here's a really important fact. We've got our Markov process on our tree. Well, if we let x1 up to xn denote the letters at the leaves of our tree, then the Markov process induces a distribution, which I'm going to denote p of t, on these letters. Okay, so given a Markov process and a tree, I end up with a distribution on letters at the leaves of the tree that depends on both the branching structure and the edge lengths in that tree. So I'm going to denote that p of t. So here's a little thought experiment then. Suppose I've got these two trees which are identical apart from the position of species 2, and then I let this edge length leading to species 2 tend to infinity in both the trees. So this is edge length L over here and in my second tree, and I'm going to let that get very long. Well, what happens is that we expect the number of changes to have occurred over that edge to tend to infinity. We're going to pick up a very large number of changes over that edge. And so what happens is that the random variable at species 2, the letter at species 2, is independent of the letters at species 1 and 3. So the distributions from these two trees converge as L tends to infinity to the same limit. So as L tends to infinity, we get the same distribution on letters at the leaves for these two trees. Let's think about what this means in terms of metrics. So I've got this distribution on letters at the leaves, and I'm going to write curly D to mean the set of all possible such distributions. Now, for a certain class of Markov process, it can be shown that the map from tree to distribution is injective. So then, given a metric on distributions, we can then pull that back to give us a metric between trees. So for example, we might take the Jensen-Shannon metric or the Hellinger metric between distributions. We pull those back to give us metrics between trees. Just as a slight aside, an issue with this is that they're very computationally expensive to compute because they involve a sum over a very large space. Okay. So let's return to our thought experiment and think about what happens to the distance between these trees as L tends to infinity. Well, we've said that the distribution associated with these two trees converges to the same limit, so the distance between these trees converges to 0. So what I've plotted here is the Jensen-Shannon distance as a function of L, so the x-axis is the edge length. And I've plotted the Jensen-Shannon distance using two different Markov models. The first is a 4-state Markov model, that's the black line, and the dotted line is actually using a 2-state approximation to this 4-state model. And you can see that both distances converge to 0 as the edge length tends to infinity. Okay, so we think about long or edge lengths tending to infinity in our trees. When we do that, if we have two leaves separated by an infinitely long edge or a very long edge, in the limit, the letters at these leaves are independent from one another. So what we can then think of doing is, if we've got a tree T and it has infinitely long edges in it, we can think about removing those edges, so breaking the tree up into a forest. And then it's natural to associate a distribution with this forest, which is just the product of the distribution from the trees, because when edges are infinitely long, we have this independence between leaves separated by infinitely long edges. So that's why we start to think about forests. Okay, it's because we can think about edges tending to a limit at infinity and obtaining a valid distribution on letters at the leaves. So just a remark, even though edges are allowed to get very long, infinitely long in our tree space, it turns out that the forests we obtain are actually only a finite distance away from the trees that we consider. They're not infinitely far away. Second remark is that spaces of forests have been considered previously. There's something called the edge product space, but geometries haven't really been put on such spaces previously. So our work can be thought of as putting a geometry on a space that's quite closely related to the edge product space of molten and steel, for example. Okay, so this slide shows contours of the Jensen-Shannon metric through a slice of tree space for n equals 4 leaves. So what I've drawn here are the contours of distance from a fixed tree. The fixed tree is at the middle of the contours. On the left-hand side, the contours were computed using a 2-state Markov process. On the right-hand side, we've got a 4-state DNA model. And what you can see is that the contours obtained are actually sort of qualitatively quite similar, even though the Markov process has changed from a 2-state to a four-state Markov process. So this is what I mentioned at the start about robustness of the metrics we obtain with respect to change in the underlying Markov processes. Okay, so we've seen how to compute metrics. However, one of the attractive things about the BHV tree space is the existence of geodesics. So how about geodesics? How might we compute those? Well, let's just think about all the trees that have the same shape. Okay, so let's fix an unweighted binary tree. I'm going to call this a topology, and think about all of the trees with that particular unweighted shape. Now they're going to be parameterized by a vector L, which lies in this positive orthant of R to n minus 1. What we can do then is equip this positive orthant with the Fisher information metric. Okay, so that's defined using the following formula. Here, P subscript L is the probability mass function of the distribution of letters at the leaves associated with the tree with these particular edge lengths. So I go from edge lengths to tree. The tree gives me a distribution on letters at the leaves, and that's the probability mass function of that distribution. So I obtain a Riemannian inner product on this positive orthant. So what I'm then able to do is solve the geodesic equation for this numerical, this Riemannian metric. So I can solve this numerically, and you get pictures like this. So this is what we call a cobweb plot. I've drawn a set of geodesics fired from this central point. So the axes here correspond to the internal edge lengths. I've got n equals 4 vertices, so I'm plotting the lengths of the two internal edge lengths on my 4 taxon tree. Okay, so I'm firing geodesics out from this central point. The difference between the left and the right is my initial tree here on the right has internal edge length 0.3 on both those edges, and here the initial edge length is 0.1. So these lines coming out, or these curves coming out, are the geodesics. The curves, sort of contours, are the contours of distance. So what you can see is that these two plots are really quite different from what you'd expect with the BHV metric. With the BHV metric, you'd get concentric circles and straight lines. Okay, so a problem with all of this is that to compute the Fisher information metric, you're doing a sum over omega to the n, so it's very computationally intensive. One way around that is to replace our discrete valued Markov process with a continuous valued Gaussian process, which approximates in some sense, the discrete valued Markov process. So we have instead a Gaussian process defined over our tree, which we can think of as being an approximation to these continuous models, and we end up with, instead of having a mass function in our Fisher information metric, we have a density function. So our mass function becomes a density function, which is the density of a normal distribution, multivariate normal distribution, with mean 0 and a particular covariance matrix. So the covariance matrix is obtained according to this formula involving the path length between leaves on our trees. Okay, so this move to a continuous Markov process is a way of speeding things up computationally. If we look at the geodesics and contours obtained, on the left we've got contours and geodesics obtained with the two-state model, and on the right we've got the continuous Markov process which approximates it. And you can see that the geodesics and contours obtained are very similar. Okay, so just to finish off then, the remaining question, which I haven't addressed at all and which Jonas will speak about then, is how to compute geodesics between trees with different bifurcating or shapes, so different topologies. So what I've done here is show geodesics for fixed tree shape, fixed topology, and I think Jonas is going to talk about really smart stuff to do with geodesics between trees with different shapes. So just to finish off then, here are some references for you to look at your leisure.
Thank you very much. So do you have any questions? No? Yes?
Just a stupid one. Why is it clear that the last sigma is positive definite?
I think it's because it's a negative definite kernel, right? It's related to negative definite kernel, I think. Schoenberg.
So yeah, it's not obvious that this is positive definite.
Yeah, but if you look at Schoenberg kernel, yeah. It was my question, actually.
Okay. Does anyone have a question too? Oh,
There's a question on the chat.
Oh, yeah, question on Zoom chat. So thank you for a very interesting talk. Can the approach also work with multivariate continuous characters / observations?
So I'm not sure if you can hear me in the room. Ah, yes, you can.
Yeah, we can hear you. You can.
Yeah, so you could certainly define a Markov process on the tree using something multivariate and obtain multivariate characters. I guess the issue would be whether the map from trees to distributions was still injective. And if it was, then this would work. But it's crucial you have an injective map so that there's a, the tree is uniquely characterized by the distribution.
Okay, so thank you very much for your presentation.
Thank you.